Hello everybody, welcome back. So today we have Dr. Sohail Iqbal from Comsat University, Islamabad campus, talking to us about classification of COVID-19 via homology of CT scans. Thank you, Sohail.
Thank you, Al-Khanan, and thank you, Henry, Sarah, and Al-Khanan for inviting me on this prestigious, stage. And today I'll be talking about classification of COVID-19 by homology of CT scan. So we have the plan is to apply topological data analysis techniques for this problem from biomedical image analysis. And it's a joint work with Farid, Talha, Imran, and Professor Nasser. So there are many kinds of data, and data has been produced from computational machines and experiments, and there are some missing, sometimes there's missing information in the data, sometimes it's higher dimensional data, and sometimes it's a noisy data. But in the last well, 1 decade, we have seen that topological approaches are very successful in analyzing the data, or we should say analyzing the shape of the data and deducing our results. And apart from their success, these techniques are very interpretable and very intuitive as compared to other machine learning techniques as well. And there are many techniques in this area, and but today I'll focus on persistent homology, which is one of the main techniques in topological data analysis. So let's have a look at some of the basics of persistent homology. The starting point is a point cloud, and from that point cloud, we construct a filtration of simplicial complexes. Well, a simplicial complex is just gluing of points, edges, two simplices, which is a triangle and a tetrahedron, etc., etc. And then we have this object. As you can see, it's a filtration of these simplicial complexes, and it's R persistent object in the category of simplicial complexes, if you like. And from this filtration, we can calculate the simplicial homology of each and every node, and the connection between these nodes. So in other words, we are talking about the functoriality of the situation, and we have another kind of object, R persistent object, in the category of vector spaces. F is basically field over here. And this object not only computes the topological feature at each and every stage, but it also gives us the connection between these topological features at different values of the filtration. So it's all very good. But there's one more problem over here. It's a bit lengthy information. So what to do with that? And with the help of some classification results, we can summarize all this information in a diagram known as the bar diagram or persistent diagram. Now let's have a look at a very simple example that depicts all this thing. So we have a point cloud at stage epsilon equal to 0. There's nothing in the h0 axis, there's nothing in the h1 axis. As you can see, that the x-axis is the birth axis and the y-axis is the death axis, and of course the death will only come after the birth, so that's why everything is above the diagram. So but you can see that in the point cloud there are many connected components. Well, you can just count them. There are many, around 20s. So if you move on, some of the connected component will die. Well, there are many techniques, there are many ways which with the help of which you can impose a simplicial complex on this point cloud. So when you do that, as you can see that now these 2 points, for example, these 2 points are now connected with an edge. So for example, one of the connected component dies over here. And similarly, you can see that many other connected component dies. So this death is kind of reported with this point on the h0 axis, and so far there's no death reported on the h1, which is counting for the loops, and or you can say that the cycles which are not the boundaries. But as you can see that there are some births over here, so let me count them, 1, 2, and now you can see that there are 2 loops in here. So there is some birth on the h1 axis, but there is no death reported, so we cannot write it down on the h1 diagram. If you move on, then you can see that there are some deaths. Okay, so these 2 components which appeared at epsilon equal to 1 stage, the filtration parameter value, and now at filtration parameter value epsilon equal to 2, those 2 die, and so that's why we can represent them. We report that death with the help of this point. Okay, so moving on. And of course, you can also see that there have been some more births of these elements, 1, 2, 3 more elements, but no deaths reported. But on the H1 axis, the connected component axis, you can also see that more connected components are disappeared. So they disappeared, and so that's why there's only one connected component now. So, and this fact is reported with the help of this point on the H1 diagram. Okay, so moving on, and there's one missing information as well. So it's 1, 2, 3, and this 4, which is kind of the shape of this data as well. So these 4 appeared, but as we move on at epsilon equal to 3, all of them kind of die, and this is once again reported as follow. And this is kind of our endpoint of the filtration parameter. So in other words, one H1, one hole remains, and we kind of report it over here at the infinity axis. So that's how we can summarize all this topological information with the help of these two axes. And of course, if you go to higher dimensions, then you will get more and more invariants. So today, with the help of one case analysis of CT scan images, I'll try to convince you that topological approaches are very, very natural for biomedical image analysis. Okay, and the thing in biomedical image analysis, the problems which we are interested in is differentiating between multiple type of cells. In cancer cells, there are many types of cells, and we want to differentiate them with the help of their topological features. Well, one of the points which I'll be emphasizing again and again throughout my talk is when a pathologist or when some radiologists look at some image, then they are really observing different shapes in that image. So that's kind of our punch line. We want to do the same thing. We want to capture what a professional medic is observing. Okay, and of course there are some other problems as well. We want to detect an image for an anomaly. For example, in a CT scan, we want to see if there is traces of COVID or not. And similarly, we can check the severity of a disease, for example, after some treatment, or to check the stage of the disease. And of course, in survival analysis, there are many studies. I don't want to name few studies, but for example, in the brain tumor, you can see the shape of the brain tumor, and you check and see, you can see the survival of the patient. Okay, so coming to the problem, we have a CT scan, computed tomography images, and there are many ways. One of the preferred ways RT-PCR, but the point with the PCR is it requires very advanced equipment and a very trained professional to deal with the equipment and to check whether a person has a COVID or not. But the CT scan is kind of relatively easily available. And but the problem is the same. We want to kind of release the burden of the radiologist to check for the traces of COVID-19 in a CT scan image. So that's kind of the main motivation behind it. So if you look at these two images, one of the images is normal, and one of the images is there are signs of COVID-19. So what are the signs? So as you can see that there are some cloudy traces. Okay, so when I say cloudy, I will try to explain in terms of intensity maps later on, but cloudy means, it's kind of ups and downs in the white region. So there are too many ups and downs, it's kind of hazy. And there are some consolidations as well, very solid white patches. And of course, one can, for example, confuse them with the, the actual normal structure of the lungs. And so that's kind of one of the aim, how we can differentiate them. And two main features, the GGOs, the ground glass opacities, and the consolidation. How can we use topological data analysis to find these two things? And of course, once again, a radiologist would observe these two things to check for the traces of COVID-19. Okay, so once again, we have these things. Now, if you look at the intensity plot, as you can see that I'm kind of releasing the intensity plot step by step. As you can see that this region over here, it's a big mountain, and it's a consolidation, and it's a white region. But on the other hand, the ground glass opacities, and you can see that their peak is not very high. But one thing is very kind of apparent in the intensity plot, that there are some saddle points over here. As you can see that there are some saddle points at each and every region of the ground glass opacities. So that's our, once again, in the intensity plot, the main observation. The consolidations are solid, big mountains, and the GGOs are kind of the saddle points. Okay, so we want to first focus on the consolidations. So let's see how we can find them. So topologically speaking, we can say that a consolidation is a kind of a tetrahedron. Okay, because it's an intensity plot, it's hollow under it, it's a surface, or a, you know, there's an associated two manifold with the intensity plot as well. Okay, and as you can see that this is non-COVID image, and the mountains, there are some mountains which are the natural structure of the lungs, but they are not very huge. They're just you know, and if you kind of vary the filtration parameter, then they will kind of disappear after some value of the filtration parameter. So the problem at hand is the intensity plots and their associated two manifolds. We want to differentiate two manifolds. Okay. Now the point is, the trouble is how we choose point cloud, which is somehow kind of close to homotopy of the underlying two manifolds. So that's our first problem, and for that we consider this feature point cloud thing. So what we do is, actually we have for example, if we have a point cloud, well, the intensity plot is also collection of pixels, so we can say that it's also a point cloud. But if we consider all of the points in the intensity plot, then it's computationally very demanding. So what we do is, we want to find out the feature point clouds which is kind of close to the homotopy of the associated manifold. Okay, so we have a point cloud, and we have some feature space, and we will see that in our case, we will take this feature space to be the intensity of the pixels. Okay, and we want this to be a compact space so that we can have finitely many open sets that can cover the whole space. And with the help of that, we can see that there is an open covering of this initial point cloud X, and we require it to be in symmetric space. So what we do is, we consider the connected components of this C of X, which is kind of the covering, and those connected components will become the vertices of my feature point cloud. And practically speaking, we just consider the centroids of those connected components, and the distance is taken between their centroids, of course. So here is our grayscale image M, and the intensity plot is, or the point cloud is I, J, and P is the intensity value at T. And we kind of consider a finite cover of this 0 to 255, which is kind of the range of gray pixel, and let W is the cover. And using this cover, we can calculate, once again, calculating their inverse, and we can calculate a covering of this point cloud. And once again, we can consider their centroids as the points of the point cloud.
Sohail, can I pause with a question from the chat? So this is a question from Ezra Miller. It's about the slide with the chest CT scan. So the question is, the chest CT slide showed comparison of the SARS-CoV-2 infection compared with normal lungs. How do they compare with other common kinds of respiratory infections, such as the flu? If you know, if this will be treated later on in the talk, then I'll wait and see.
Okay, unfortunately, it's a kind of 1.5-year-old work, so I'll not be kind of dealing with that over here. But yes, that's a very important question, and it's a kind of I'll not be presenting it over here, but it's doable.
When you say doable, it's also doable to distinguish COVID from
Yeah.
other
Yeah.
respiratory?
Other. Yes. Yes.
I see. And visually, does it look different to the eye?
Yes. Yes. There are some biological reasons, and of course, we can kind of model them.
Okay, thank you.
Well, once again, the point is the same. If a professional medic can do that, we can try to. I mean, he's just observing a surface, the intensity plot, and all we are trying to do is try to see what they are observing and try to extract that feature.
Makes sense. Okay, thanks.
Yep. Okay, and then from here we have this Vittori's-Rix filtration, and the upper bound is taken to be 500. That's our infinity, and using this filtration, we can calculate the persistent diagram. So let's have a look at one of the example. So there are different stages of the intensity. So in the first stage, you can see that there are some points, and the next stage, you can see that the connected components are there, and the points which are the centroids of these connected components, you can see them. And if we move to the next stage, you can observe some more points, and the next stage, and of course the next stage. Well, you combine if you combine all of these points in one picture, then we get something like that. So we have in the background we have a CT scan image, and in the foreground you can see that the points and the points are different in color at different stages of the intensity plot, or you can say that corresponding to different open sets of the covering, W1, W2, up to so on. And of course, if you have this thing, you can calculate different persistent diagrams in different dimension, H0, H1, and H2. So that's kind of one pipeline of our construction. Okay, so we have ground glass opacities. So that's our next problem. So with the consolidation, the tetrahedron construction kind of works. But when you talk about the ground glass opacities, so they are kind of connected peaks, but they are not going all the way down to the ground floor. They are just connected with each other. The peaks are connected with each other, and they are creating more and more saddle points. So, and unfortunately, these persistent diagrams, which we just constructed, cannot kind of detect these things. And these are kind of the initial stages of a COVID. So these are more common in a CT scan. So that's why these are more important. And in the second last slide, I'll try to show that the next strategy is much more, gives us much more high percentile of results. Because in most of the images there are more ground glass opacities than as compared to consolidations. So that's the next thing. And once again, as you can see that there are some saddle points over here. And to deal with this or to detect these saddle points, we are going to use low star filtration. And in the low star filtration, we have we associate with each and every vertex, we have some function. For example, in our case, we can say that it's the intensity of that pixel. And then we extend it using linearity to the whole simplicial complex. And then each node of that filtration of the simplicial complex is defined in the following way. Sigma belongs to delta if the maximum of f of b is less than or equal to epsilon. So that's how we can construct each and every node of the filtration of the simplicial complexes. Okay, and the connectivity is taken to 8 neighbors or less if it's at the corner. So the point is we are taking too many vertices, but it's much more efficient to calculate low star filtration because everything is kind of linear in this situation. Okay, so burst times are local minimums, and the depth times are the saddle points, and with that thing, of course, in a zero-dimensional persistent diagram. So having said that, let's see what we have got so far. So we have saddle points over here in this picture in the intensity plot, and we have these peaks which can be detected using the persistent diagrams H0, H1, and H2, and particularly H2 is at least theoretically, H2 is much more important because at the end we will see that H1, H2, and H0, they are kind of equally performing. features. But intuitively, H2 seems to be much more relevant to this thing as well. But okay, so that's kind of our the situation so far. So we have a COVID image and a non-COVID image, and as you can see that their persistent diagrams. So H0 is different, H1 is different, H2 is different, and low star filtration, the zero-dimensional persistent diagram, is obviously also different, as you can see. Okay, so what do we have? So we started from a collection of images, COVID and non-COVID images, and now we have a collection of persistent diagrams. Okay, so and of course the ultimate goal is to develop a model that can differentiate between these two persistent diagrams. So in other words, what do we do? What we want to do right now is, or what we need is some sort of tool that can help us in doing statistics on this space of persistent diagram. Okay, so for that we are going to use a kernel on the space of persistent diagram. Okay, and I'm going to use the study of Sohail Anirudh et al. And in that study, what we do is we start from a persistent diagram and we approximate it with a two-dimensional PDF with a support from 01 to 01, but that's kind of not important over here. So what do we do? Let's say we have this persistent diagram, okay, and with the help of this persistent diagram, I can calculate this sum of Dirac delta functions. Okay, so let me try to plot it. It's not going to be a very good plot, but let me try it anyways. So let's see, this is the z-axis. Okay, and the z-axis corresponding to each and every point, I consider this Dirac delta distribution. Okay, so let's say this is the point P1, P2, P3, P4, and P5, and P6. And corresponding to each and every point, I have this Dirac delta distribution. And for those who are getting confused, it's these lines are going towards the z-axis, and this is my z-axis. Okay, and once you have that, then you are already in some Hilbert space. That's exactly our requirement. If you are in a Hilbert space, then you can do all of the statistical operations. But the problem with that is this this kind of conversion is not very robust to changes in the persistent diagram. So what we do is we solve a heat diffusion problem. Okay, and with this the sum of Dirac delta functions as the initial conditions, and it's a Dirichlet problem, and the boundary is kind of the diagonal of this persistent diagram. Okay, so the solution of this problem is going to be, so one stage of the solution is going to be, as you can see over here, and let me just try to sketch the diagonal over here. So this is the diagonal. Okay, now what is really happening? So these persistent points, the points in the persistent diagram are the heat sources, and as you move on in time, the heat is kind of distributing in the environment, and at time t is equal to sigma, you will have a diagram like that, and it's kind of now more stable, and it's an element of a Hilbert space, L2 of sigma. So that seems like we have achieved our goal. So we have mapped persistent diagram into a Hilbert space. But that's not really happening over here, because it's kind of computationally very demanding, because the space which we are in right now is has a metric, Fisher-Rao metric, and it's very computationally demanding metric. And the solution to this problem is basically posed by initially it was used by Srivastava et al, but Sohail Anirudh et al used it in their study. So the solution to the problem is to work in a square root framework. As you can see that there is a square over here. So we are in a square root framework. And when you do that, then you are mapping basically each and every persistent diagram on a Hilbert sphere. Okay, so and on that Hilbert sphere, we can actually calculate, there is a closed form expression for each and every operation required for our analysis. Okay, so that's the general picture over here. So we started from a persistent diagram, we mapped onto a two-dimensional PDF, and from there we mapped onto a Hilbert space. okay, and using the square root framework. So that's our framework. Okay, let me wait for the finish. Okay, so, and that's the closed form of for example, the distance between the two persistent diagrams, and we can also calculate the geodesic, and we can the exponential map, etc., etc. So everything has a closed form, and we are going to use this closed form for a very computationally effective approach. Okay, so here's the summary. We started from images, we got the persistent diagrams, and from there we have used this approach by Rushil and Anurad et al., where we converted each and every PD to two-dimensional PDF, and from there we are on a Hilbert space. So we are on a Hilbert space. Each and every image is on a Hilbert space. So what we do is we study separately. For H0, we study it separately. The pipeline is different, and for H1 we study it separately, and for H2 and similarly for LSF persistent diagram. Okay, so here is the once again the summary of the things. We started from here. After the quantization, we got this point cloud, and using the Vittori's-Gibbs filtration, we got this persistent diagrams, and from here we map them onto Hilbert space. And on the Hilbert space, these conversions so far, the maps, the points on the Hilbert space, they're very high dimensional. So what we do is we use the principal geodesic analysis to reduce the dimension of each and every feature vector. Okay, so after doing that, we get each and every feature vector is basically a length of 4800. Okay, so each image, okay, is now converted into a feature vector of length 48. And from here, we can use, for example, a sport vector machine. We use sport vector machine, but later on we used some other, for example, artificial neural networks as well. And so they performed well as well. But my point is, we can use a very basic machine learning tool as SVM as well, and the results are very, good. Okay, so so here's the summary of the comparison. So our approach is topological approach, and not only these technique performed well, but as you can see that the variance is relatively low as compared to other 99s. Okay, so as you can see that whenever there's 99, the variance is a bit higher as compared to our approach, and these are all very advanced neural network algorithms, and this topological approach is performing very well as compared to this thing. Okay, and in the last stage, let's compare different features. For example, with the help of, if I just consider the H0, then here is the accuracy, precision, and recall, specificity, and other F1 scores. And similarly, you can see that H1 is performing not very well, and H2 is also performing not very well. But so we think that, and later on we tested on other data sets as well, and the interpretation is here that the data set is not very for example, the disease is not at a very advanced stage, most of the images. So as you can see that the consolidations which we planned that H2 should kind of detect, but there were not so many consolidations in our images. So that's why H2 is, in this data set, H2 is not performing very well, but in general, it performs, if you consider only the images with consolidation, that it performs very well. So the point is, it's kind of very well-balanced thing. And in the last, we have low star filtration. And the output, as you can see, that it's very outstanding. But the point is, most of the images, they were at the initial stages of the disease. And so there were kind of the ground glass opacities and not so many consolidations. And ground glass opacities, we planned that the low star filtration should catch it. And so that's why, the LSF is performing really well in this case. But there are some images where the LSF is not performing well. So here is one image in which the low star filter, if I just consider the PD of the low star filtration, then it could not detect that this is a COVID image. So the reason is you can see that not so many saddle points over here. So just peaks, and this is the COVID region. This is the COVID region, and over here you can see that it's kind of very close to the wall, and it's close to the wall. And some features like H0, H1, and H2, they could pick it up, but the low star filtration, due to the lack of saddle points over here, could not pick it. So that's why all of these features, and we have tested on some other data sets as well. So all of these features are kind of required for each and every stage of the disease.
So I have two questions, two good questions from the chat, if that's okay. So the first question is, could this method be used to detect different variants of COVID-19?
Well, we have not seen it, and to be very honest, I have not seen any data set for Omicron or Delta or other variants.
Interesting.
If I see it, then we'll see.
Before I ask the next question, so just to follow up to that, do you know from the time that this data was collected, was it roughly all one variant?
Yeah, it was collected very early, like early of 2020s in Brazil, Sao Paulo. So
okay. And
they added some more images in the data. Sorry, so they added some more images in the data, but for those images, that's a very good question. I'll have a look at that. So the variants.
And the second question is, have you considered using a similar approach to perform a segmentation of the pathological tissue, for example, segment lesions, classifying them as GGO, consolidation, or honeycombing?
Well, segmentation is a difficult problem, and but there are some studies. I use them, but so there are some studies which are kind of finding the cycles, the inverse thing. So there are many papers. I can send you the link of these papers where they kind of find the topologically important regions and find their inverse images in the representative cycles, and from those cycles you can try to find out the important regions in the image. So that's something which is done so far, but segmentation just from the topological approach, I have not seen it. I have not done it. Well, you can combine them with other neural networks as well. But
well, thank you for the answer. Okay, please continue.
Okay, so now the interpretability is kind of the key thing of our study. You can see 3 COVID images, and you can also see that the COVID regions. So over here, this is the COVID region, this is the COVID region, and over here, this is the COVID region. And if you kind of hide the COVID regions, then our model exactly tells us that this is a non-COVID image. And if you put this box on some other region apart from the COVID, then once again, our model can say that, no, this is a COVID image. And similarly, there are some other random approaches as well. So that's the interpretability of our result. And of course, apart from that thing, we can also tell why the model is detecting due to which topological feature. So thank you very much. That's all from me.
Thank you. Okay, so before we get to questions, I'd ask everybody to please unmute themselves and applaud the speaker. Okay, so we have plenty of time for questions. So as we have a lot of people in the audience, maybe if you could raise your hand, or there's functionality for that. Or I guess just unmute yourself.
Yep.
We have a question in the chat. So ground glass opacity, the manifestation of COVID-19 in the lungs, may be caused by other ailments such as pneumonia that are unrelated. Are there any salient differences between the COVID-19 ground glass opacity and other types that your model can identify, or is it picking up on all the ground glass opacities in CT scans?
it's just picking up all of the ground glass opacities, but with the other lung infections, there are some locations which are important. So yes, if you try to develop a model for that, then not exactly this thing, but you can try to see that the regions in which they are appearing. So we can do that, but not in here.
So we have another question. How do you compute the centroids of the covers?
Yeah, so what I mean is
centroids
the individual sets in the covers is what I mean.
Okay. Okay. So you kind of divide the interval 0 to 255 into regions, and then you kind of consider only that part of the surface which is in that region. Okay, and ignore all of the other parts. And then
Right.
if you talk about in Python and MATLAB, there are some commands which can compute the connected components.
So I mean, so if you do that, then you probably get a set that's disconnected.
Yes, yes, of course. That's the point. That's the point. At each and every region, in each and every block, we want to see that which are the connected components which are important features.
I see. And so then in each of those connected components, you compute the centroid.
Yeah, yeah. So the idea is if you can see my screen. So the idea is try to find out points at each and every stage. So for example, if there is a consolidation, then eventually it's going like this, a peak. It's going like a peak. At each and every stage, you are getting the pixels, and and eventually it's getting a peak. So that can detect the things.
Okay, thank you.
We have two more questions, so I'll just go in order. Did you consider using persistent images in order to transform the persistence diagrams to feature space?
In order to transform persistent diagram to?
Yeah, to vectorize the persistence diagrams, did you consider using persistence images rather than the kernel method?
Okay, so persistent images and the thing which we discussed today is kind of similar. It's kind of similar, but 2 years ago, I'm new into this area. So 2 years ago, I didn't know about persistent images. So that's why I didn't use it. So yeah, that's my excuse for not using it.
So the next question is, can this be used to measure the severity of the infection in the lungs, perhaps by computing the volume of the lungs impacted by COVID?
We don't have to actually use the geometrical approach of calculating the volume. The filtration can take care of it. So if it's a big area of infection, then a larger value of the parameter will hold that feature. So we don't really have to calculate some volume or other things to see the severity. The filtration can take care of all of these things.
Henry has a question.
Sure, I had a question about how did you learn all this about COVID? Did you sort of learn it on your own as you went, or are you working with folks who helped teach you some things about the lungs, etc.?
Well, that's a hard problem. That's a hard part of any problem. The initial stage where you try to connect different pathologists or radiologists and try to understand the things. But all of these things started from an online lecture where a doctor was trying to explain the difference between the COVID and non-COVID image. And I was looking at some, accidentally I was looking at this data set, and I was like, okay, I can do that. So, and then, with the help of course, collaborators and all of this thing, So cool. Yes, so in short, so the new problems which we are working on is, that's very hard problem. Try to understand, the biology behind the things. So, so ask my students, they are always in trouble in the initial stages of the project.
Thank you.
So we have another question. So thank you for your talk. So that's not a question, but can this TDA approach be more robust against noise or perform better than other, say, CNN models for unseen data?
Well, I don't want to, I have used these CNNs, but not in connection with TDA in a serious study. But yes, people are doing that. So unfortunately, I cannot say more on this thing. But you can do is, what you can do is the same thing which I was talking about earlier, that you can try to find out using these topological approaches, you can find out the important cycles in the image, and that will give us, the relevant or more important regions in the image. And then you can, in the CNN, you can only focus on those regions, and that will improve the time and the other things as well. So that's people are doing.
I have a question. Could you speak actually to the, this is one of the behind the scenes questions, about how much time does the pipeline take to run?
Oh, that's a variable answer, because more and more libraries are coming, and for example, now we have Ripser Parallel and Giotto and other things, so we can run our programs on Google Colab. So it depends. So for example, nowadays we are trying to compute a problem, and for each and every image it's taking 30 minutes, not acceptable. So the next thing what we can do is only consider the appropriate, so the thing which we did in this study, so only consider the appropriate point cloud. So choosing a point cloud is, and the computational limits is kind of it's all about balance, how to choose a point cloud which is kind of related to the homotopy type of the underlying the manifold or the associated manifold, and the computational limits. So so it's a balance. It's a tricky thing. So in the beginning, it was a trouble. So we started from JavaPlex, and JavaPlex could only handle 500, 600, points. And now the Ripser Parallel or the Giotto, they could handle 5,000, 6,000, but they take too much time. And then, it's all about managing. But yes.
Makes sense. I guess I have another question, not quite necessarily related, which is maybe a question of visualization. So once you transform all these images into your vectors of length 4,800, so I guess the question is, do you think, I don't know, if you did some dimensionality reduction, you could visualize something about whether or not topologically, are all the COVID cases topologically similar? Because you seem to suggest there are some COVID cases in which certain topological features are present and not others. Sort of, how does the topological variation, if you could visualize the vectorization, could it tell you something about what the shape of the whole data looks like? Is there something? Yeah.
Yes. Unfortunately, I couldn't present that diagram over here, but we plot this TSNE plot, and you could see that there are clearly two connected components, but not more than that. Not more than that. So there was a TSNE plot.
Interesting. Another question, we have two questions. So another question in the chat, what is the choice behind the feature vector of length 4,800? I guess, how did you come to that
It's
number?
1,200. So it's 1,200 before. So each feature was 1,200, and then so H0, H1, H2, and LSF. So why 1,200? Once again, it was yes. To be very honest, it was hit and trial. So we reduced it further, and it was not performing, and we increased it, and it was computationally demanding. So the whole thing takes around 256 GB RAM to load all of these features. So initially, and then you reduce the dimensionality. So you want to balance the things at each and every stage with the computational limits.
Okay, well, that answers it for me. I mean, Mohammed, you can write in the chat if it answers it for you. We have another question from Henry.
Yeah, so Sohail, I really liked your images where you take a 2D image, say a grayscale image, and then you turned it into a 3D surface. And I think, especially for newcomers to our field, this is a wonderful visualization to explain what's going on. I was curious, how did you create that those 3D visualizations of 2D images? Did you write this code yourself or with some of your students, or yeah?
Oh, no. It's just ImageJ. So there's a very good package of ImageJ where you can visualize all these things. It's there is a well, you can also do that in any Python or MATLAB, but there's an important feature in ImageJ where you can smooth the surface. Otherwise, it's very sharp. So it's very well used package for computer science people.
Thanks. I appreciate it.
So thanks to Talha, my collaborator. He's from computer science, and he tells me all these tricks.
And any other questions for our speaker? Okay, if not, then I'll end the recording.